Hello and welcome to our first screencast for Bioinformatics 525, Foundations of Bioinformatics and Systems Biology. This is a 3-module course. Our first module, Introduction to Bioinformatics, begins on January 12th. My name is Dr. Barry Grant. I'm an assistant professor in Bioinformatics and Computational Medicine, and I am your course director. In addition to lectures from myself, we'll have one guest lecture from Dr. Ryan Mills, shown in the middle here, on genome informatics. You'll also get the opportunity to interact extensively with our GSI, our graduate student instructor, Hongyang Yi, who's in our Bioinformatics Ph.D. program. Now, if you have any questions or concerns, both Hongyang and I are here to help. If you can't catch us directly in class or in labs, then please do initiate contact by email, which is typically the best way to get in touch with us. We will meet twice a week in Palmer Commons. Our lectures are in room 2062 in Palmer on Central Campus, and they're Tuesdays from 2:30 to 4 p.m. Labs are on either Thursday or Friday of the same week, commencing at either 2:30 for the Thursday labs or 10:30 a.m. for the Friday labs, also in Palmer Commons, second floor, room 2036. Now, you should have registered for just one lab session, either the Thursday or Friday, and then you'll attend that session throughout the course. Now, labs really are the most fun part of this course because it's where we'll do most of our hands-on explorations, both individually and in small groups. Lecture slides and video recordings, lab handouts, background reading material, etc. will all be posted on our course website. Our course website is open. It's going to be up there for as long as we have an interest in this material, so it's going to be up there for you to access after the course is finished. Now, if you're watching this video, this screencast, then you've likely found the website already. If not, you can find it by going to tinyurl.com/bioinf525-w16. Okay, and I'll put that in the email announcements to you as well. So again, please let the GSI, that's Hongyang, or myself, know if you have any trouble accessing any of this material. Okay. So what is bioinformatics, or what does it mean to you? Because as we'll see in class when we discuss a lot of different definitions together, this term bioinformatics can mean different things to different people. So we'll cover some of the most widely accepted definitions in class, from simple, concise definitions like this one, where we're talking about the computational management and analysis of biological data, through to more extensive definitions that spell out core approaches and major application areas that we'll cover in class. So what do we mean here with this simple definition? First of all, the computational management. Well, what we mean there is really the collection, archiving, organization, and analysis via computers. Okay, this is the application of computers to biology, right? As well as the analysis of biological data. So really, bioinformatics, and this is at the core of all these definitions, it's a hybrid of biology and computer science. Or even more simply, you can think of it as the application, as computer-aided biology, right? It's useful to think of bioinformatics as a wide subject, as wide as biology itself, and the application of computers to understanding that type of data. So for the purposes of our introductory course here, we'll take a much more hands-on approach to learning bioinformatics than we would in some of our other courses in this program. And I will really try and encourage you to think of bioinformatics as this process of extracting biological knowledge from large-scale data, biomolecular data, biomedical data in particular. So really what we're aiming to do here with bioinformatics is to bridge this gap between data, we can have a lot of data for example, and knowledge. So it's important in this context to remember that bioinformatics is neither tied to a particular type of experimental data nor to a particular biological application. Indeed, the data itself may come from any of today's high throughput multitude of bioanalytical analytical methods that are becoming available, such as next generation sequencing, nuclear magnetic resonance, mass spectrometry, medical imaging, etc. And the knowledge as well can be varied, right? The identity of new disease biomarkers, characterizing the evolutionary or phylogenetic relationships of some important biological macromolecules, obtaining quantitative structure-activity relationship profiles for prospective drug molecules, predicting an unknown biomolecular structure, potential inhibitors or activators, or obtaining even system-wide understanding of a particular disease state, for example. Similarly, the tasks involved in bioinformatics range from simply organizing complicated heterogeneous data for future use through to sophisticated analysis, visualization, and sharing of the data and indeed the results derived from it. So bioinformatics is truly an interdisciplinary research area requiring understanding of biology, mathematics, statistics, and of course informatics, which is really the computer science. Okay. So our objective in Module 1 is to provide an introduction to the common practices of bioinformatics, as well as this practical guide or hands-on guide to using major bioinformatics databases and algorithms. We'll begin with our first lecture with the what, where, why, and how of bioinformatics. That's what is bioinformatics? Where did it really come from? Why do we need it? And how do we do it? Right. So what are the major research challenges and kind of approaches that we take with this relatively new field of bioinformatics? We'll then delve into some of the most widely used bioinformatics resources. That's for the first lecture, we'll concentrate on databases and associated tools for macromolecular data. That's primarily DNA, RNA, and protein data, and how we can use these for our research, right? In our second lecture, we'll begin going into some of the details of the algorithms and tools that underlie this process of sequence alignment and database searching that we'll touch on in Lecture 1, but really delve much more deeply into in Lecture 2. So these are core skills of bioinformatics, right? How do we get useful data out of these large sequence repositories, and how do we get the computer to work for us, right? But also because it's a way for me to introduce important bioinformatics algorithms. These methods that we'll cover here underpin quite a lot of what we do in bioinformatics, from analysis of genomes through to phylogenetic reconstruction and the structural bioinformatics. So this lecture will give me a way to begin to describe to you, or to let you think about how to get computers or computer algorithms, these recipes or protocols that will tell the computer to do complicated tasks for us, right? These tasks may on the surface, such as this sequence alignment approach, seem intractable in many ways when you really sketch it out fully. And then we'll cover how we can take shortcuts, or what we call heuristic approaches, to make this analysis much more feasible on a large scale. Okay, so this is important because bioinformatics is frequently about large scale analysis, not just analysis of a single gene or a single protein or a single sequence, for example, but perhaps an entire set of genomes or an entire set of proteomes, for example. So in our third lecture in the series of this first module, I'll take you into the fascinating area of structural bioinformatics. This is the domain in which I and my lab spend most of our research time. We'll talk about biomolecular sequence, structure-function relationships, and how we can use computational approaches, bioinformatics methods, to really aid in deciphering these all-important relationships in healthy and diseased states. And this helps us reveal kind of fundamental mechanisms of how biomolecules actually work at a very detailed level, and how we can use these methods to aid with drug discovery and the design of new proteins. Finally, for Module 1, we'll conclude with a guest lecture, an associated lab from Ryan Mills, on genome informatics. And this will take us through kind of high throughput sequencing, applications, and analytical methods, really the application of bioinformatics to genomics and some of the major computational approaches that are indispensable for the current genome scale analysis that is so prevalent today. Now, I would argue that the most important component of this course is not the lectures themselves or the screencasts or even the reading material and the quizzes that we'll do together, but it's really the hands-on components, both in class, but more extensively in the lab sessions. So it's very important that you remember to bring your laptop to class, and I would encourage you to preferably have it charged up and ready to go. And please have an updated a browser such as Safari or Firefox or Chrome or web browser installed on your own laptop. Now, the only other requirement at this stage is that I ask you to please take a moment to fill out our very short pre-course questionnaire, and you can see the link here. It's also on our course website, and it'll be in your course announcement. This questionnaire is important because it helps me tailor the content that I'll deliver for you, and hopefully help me also to justify the existence of this course to my department by learning a little bit about your background and your interests. Okay, so deciding what to include in a course like this is not always easy. We have whole courses devoted to each of the topics that we might introduce in a single lecture here, and selecting the topics to cover is a bit like being a tourist guide to a country, maybe Ireland where I'm from, or indeed any country. It's just not feasible or wise to attempt to cover everything of interest. Okay, so information really needs to be carefully selected so that it helps newcomers orient themselves, outlines how you survive in a new country or a new field, covers a few highlights that give you a real flavor for what is possible in that country or in a field such as bioinformatics. So personally, I want to welcome you to the course. I hope that you'll enjoy your visit and that we'll whet your appetite and you'll come back for more. Okay, so thank you very much for watching, and I'll see you in class.